Hello dear students, I am Priyanka Sharma, back with the biology of class 12, chapter number 3 we have started with, and in that we have seen with human reproductive system, male and female reproductive system we have seen with, okay. Then we have started with the topic that is gametogenesis in the previous lecture, and in gametogenesis also we have seen with the spermatogenesis in the previous video. See, in spermatogenesis we have seen that how a sperm is formed. Just a quick revision of the things that we have seen in previous video, okay. So over here spermatogenesis is a process of formation of sperm, and in that process of formation of sperms it has certain events formed. That events comprises primary spermatocytes formation, secondary spermatocytes formation, spermid formation, and then spermatozoa, or you can say sperm formation, which is somewhat this shape we have seen with. At last the sperm formation. So to form this sperm we have seen with certain events. That events comprises of primary spermatocyte formation, which is being formed by mitotic division, okay. Once the mitotic division leads to formation of primary spermatocytes, then meiosis division, meiosis 1 division divides and it forms the next one, that is secondary spermatocytes we have seen. The secondary spermatocytes again divides mitotically, and that is meiosis phase 2 to form spermatids, okay. This spermatids further forms and divides again mitotically to form haploid cells with 23 chromosome, okay. 4 it forms and 23 chromosomes to form spermatozoa, that is spermatids. So this is how the whole process of spermatogenesis we have seen with, and in this process we have seen with the term named spermiogenesis. Now spermiogenesis is process of formation of spermatids to spermatozoa, or formation from spermatid to spermatozoa or sperm is called as spermiogenesis, okay. This was all regarding sperm formation, and we have also seen with hormones involved in formation of this, the hormones which are affecting the growth of this gametogenesis as well as spermatogenesis we have seen with, okay. In male we have seen that certain hormone, that is LH, growth stimulating hormone, follicle stimulating hormone, these hormones were involved in affecting, they were just involved in affecting the Sertoli cells, okay, Sertoli cells, then Leydig cells also, okay, Leydig, it is Leydig cells. These Leydig cells were involved in secretion of androgens, okay. Androgens were being secreted by the Leydig cells. This all things we have seen in the previous video. Please always, when you start reading with any topic, start from revision parts of the previous thing that you have studied. Once you revise or recall by yourself that this thing I have studied in the previous video and all, then it will help you to recall the next part. आपको immediately समझ में आ जाएगा कि next thing I have to study this. Always have revision practice while studying. Once you complete spermatogenesis, then revise by yourself. Start saying with yourself, talk with yourself and explain this. Then it would be easier for you all. Then we have seen with the structure of sperm. I would like to show you how actually the sperm looks, okay. In that we will say with over here, this is how the human sperm looks. Sperm means seed, and then zoons means and in animal, okay. Now we can see over here the next one. See, this is the structure of sperm, having a sperm layer on the head part. See, sperm I have told you it is divided into 4 main layers: head, neck, middle piece and a tail. These are the 4 things which are comprised in a sperm. Now this part is called as head, this region is called as neck, and this part you can see mitochondria, centrioles present over here, 2 centrioles. These are helping it as a digestive enzyme, okay. Mitochondria helps in swimming, it provides motility, okay. Then a tail part, okay. This Sperm motility is essential for fertilization, and motility is being provided by, I have told you, the middle layer. Okay. The next thing we'll see that, middle piece possesses numerous mitochondria which produces energy for movement of the tail. For movement, this particular part, middle piece, is responsible. Next we'll see with the, it is the same, the structure of it. Here you can see acrosome cap, nucleus present in the head part, mitochondria and a tail, and this, the region between tail and the middle piece, this filament like structure is known as axial filament, which is moving towards the top. So seminal, let us say that seminal plasma along with the sperm constitute the semen. Next thing is the function of male accessory gland. We have seen in male reproductive system, that is testis we have seen, accessory ducts, accessory gland we have seen with. Now here you can see that the functions of male accessory ducts and gland is to maintain the testicular hormone, which is androgen. Okay, that is being maintained by this particular part. Next thing, if you see regarding the parts that I have already discussed, acrosome. Okay, now for today's lecture we'll start with the next one, that is oogenesis. So we have seen with the male part, that is spermatogenesis, that was regarding male, and oogenesis is a process in which ovum formation takes place. Okay, ovum. Next we'll see in this is how ovum, see you can see over here, the process of formation of mature female gamete is called as oogenesis. Okay, then oogenesis is initiated usually during the embryonic development. Okay, and it is being generated from the mother cell. See, the overall process it comprises, that is from mother cell or mother gamete cell, oogonia are formed, and from oogonia development of primary oocytes will see. Further I will show you the steps, but just have a quick, you can say quick steps that are involved in this. See, the steps involved in this are almost similar to the spermatogonia and all we have seen with, but slight difference over here we will see with in further part, that is this oogonia will further divide to form primary oocytes. Okay, this primary oocytes further it goes for the mitotic division and meiosis division, and it will form primary oocytes. This primary oocytes will again further divide into some cell, and it will be covered with the layer which will be called as primary follicular or primary Graafian, it is also called as. And then further if you see with this primary follicular part will divide to form secondary, and secondary will divide to form tertiary. You are getting with this point, primary, secondary and tertiary. Then this primary, from primary to tertiary, the layer, the changes that we see will lead to formation of secondary oocytes. Okay, and from the secondary oocyte it will form mature Graafian follicle. This is the short event procedure I have told you, events that is occurring in oogenesis. If you compare with the terms, in spermatogenesis we have seen with primary spermatocytes and secondary spermatocytes, but the difference over here is formation of primary follicular and secondary follicular as well as tertiary follicular. This is the only difference. The main thing, that is primary and secondary are the same one. Next we will see with in this, see over here prophase 1 process is occurring of meiosis, whereas in previously for spermatogenesis we have seen that mitosis. Okay, mitosis and meiosis 1 phase was seen in male when we were starting with the spermatogenesis. Okay, and in this we are having only this phase, that is prophase 1 of meiosis. Okay, next we will see again. See over here I have told you primary follicular formation. I hope this diagram is not clearer that much, but see with the point that I am telling. See, primary follicular formation, oocytes formation and secondary follicle. This secondary follicular will develop to form it will form Graafian follicle, okay, the developed part. And again, if you see with the corpus luteum formation, and at last the mature ovum is being released with the help of ovulation process. See these small things that you see over here, these are called as primary follicle, okay. This primary follicle forming to the secondary follicle, जब secondary follicle form होता है, then you will see the first polar body formation, okay, and then further you will see second polar body formation also. I will show you the steps in this. First you remember with the step is formation of primary oocytes, okay. This primary oocytes, okay, I will tell you again, this primary oocytes are formed as a result of division of cells, that is prophase 1 of meiosis. पहले ये form हो गया, then after this formation of primary follicular, or you can say primary Graafian follicular formation, okay, or follicular or granulosa formation, okay, granulosa cell formation. Next is see over here, it is written follicle at this stage develop to form primary follicle. This stage is formed when the primary follicle divides at meiosis prophase 1. जब meiosis prophase में होता है, that time primary follicles are being formed. Next, if you see primary follicle and this, when the primary follicle formation takes place, then a cavity named antrium appears within the membrane of granulosa, okay, and that follicular cavity will increase in size. See, you can see over here, this size is small and the sizes are being increasing. This increase in cell or size of a cell leads to development of a mature Graafian follicle. See over here, the sizes are increasing. First they were smaller in size, as they mature then it forms, the size increases, again the size is increasing. The primary follicular are in this shape, and if you see with the secondary follicular, they are of larger in size. That is because of antrium, okay. See, as the Graafian follicular expands, the stomatal cells surrounding the membrane of granulosa becomes condensed and forms a cavity known as theca interna. See, once the theca interna is formed, if you see with the structure over here, this line is having cells grown at the periphery and then growth of cells. Now this covering, यहाँ पे क्या है कि as Graafian follicle expand, the stomatal cells surrounding the membrane of granulosa becomes condensed. जैसे जैसे cell growth होता है, the cell becomes condensed and forms a covering called as theca interna. Same way, this is internal part, अंदर का जो part है that is covered by theca interna. Same way, outer part is called as theca outerna. Here you can see externa or outerna. External यानी बाहर से, and अंदर से it is from inside interna. I hope you are clear with this. We are seeing with primary follicular to secondary follicular development, the changes that is occurring from this to this particular part. Before this, it has primary granulosa, or you can say oocytes. This is the first stage. Then it goes to the second stage formation, primary granulosa, or it is also called as primary follicular. This primary follicular cells will form to secondary follicular cells, or it is also called as secondary granulosa. See, these are just the covering part. The cells when they are dividing meiotically, that is in prophase 1, then these are the changes that is being seen. Okay, next thing, if you see, I will write the steps later on. First see with the diagram what are the changes occurring. See over here, now the follicles are called as secondary follicle. Then this cells of theca interna later secretes a hormone called as estrogen. So estrogen is a female hormone, they are being released during formation of ovarian follicle. See, follicle means what? I hope follicle terms आपको पता होगा. Follicle is a tough covering or it is a dense covering to the ovarian walls. Okay, this dense covering is seen during oogenesis process, or you can say ovum process. In the stage of ovum process, the follicles are being developed. Okay, then if you see with the next part, see here the diagrammatic representation. Primary oocytes will form the one polar body and a secondary oocyte, and in this stage they have development of primary, I am just writing in short form, primary granulosa or primary follicular, plus it also forms secondary granulosa and follicular. Okay, from this stage to this stage, in between it has formation of primary granulosa or primary follicular cells, and the second one, that is secondary granular or secondary follicular cells. I hope you are clear with this. यहाँ से यहाँ तक बनने में it has two main events occurring. That event are formation of primary granulosa or primary follicular cell development, and the second stage that it is having, that is secondary granulosa or secondary follicular cell development. Then when the secondary oocytes are being developed, it has development of first polar body. Okay, then this first polar body is haploid in number. Okay, next if you see within this, see over here, the secondary oocytes having a first polar body again divides mitotically to produce ovum and a secondary polar body. This is how this secondary polar body again generated from the first polar body has three polar body formation. You can see over here. I'll rub this part for you so that it would be clearer for you. See, the secondary oocytes retains the bulk of cytoplasm, that is once it has nutrient of rich primary oocyte, then the second mitotic stage begins but stops at metaphase 2. When ये stage पर जब stop होता है, then it has formation of ovum. Along with it, it has secondary polar body formation, and from the first polar body which was formed during the secondary oocytes, it released the two again polar body. So total we have formation of three polar bodies. I hope you are clear with the topic. ये बहुत ही simple है, सिर्फ आपको primary secondary, how primary follicular cells are formed, how secondary follicular cells are formed. That you have to see in ovulation process or oogenesis process, and how the cells are dividing. They are dividing mitotically or meiotically. If they are dividing meiotically, then which phase they are having? In starting we have seen that in prophase 1 the reaction was starting, and then it is ending up at metaphase 2. Next we will see in this. See, the secondary follicular, it leads to further change into mature Graafian follicle, or it is also called as tertiary follicle. See with this part, I have told you primary secondary याद रखना है आपको, और लास्ट में जो Graafian follicle, the mature Graafian follicle forms, and that mature Graafian follicle is also called as tertiary follicle. Okay. This was all regarding, see this is how the actual process formation During fertilization part, see this is egg part. Egg में से polar body is there, secondary oocytes formation, then it involves meiosis 2. After fertilization, meiosis, primary cell and oogonium formation. From oogonium, primary oocytes formation, secondary oocyte formation and polar body formation. This is how the process is being formed in oogenesis. oogenesis process में पहले जो oogonia होता है, that is immature part, it leads to formation of mature part. Okay, mature कैसे होता है? And in maturation event it has formation of secondary and primary formation. I hope you are clear with this. And this is how, see, in human male the oogonia starts the cell division during the embryonic development and stops at the process, that is at prophase 1, starting phase, or mitosis is the end phase. See, this is the step, ovulation process and the structure of ovum. The shed, see, ovum is a shed from ovary and is mature and arrested in metaphase 2 of the maturation, that is division in meiosis 2. This is how the egg looks or ovum looks. Okay, that's all for the lecture. In the next part we will see regarding the menstrual cycle. Thank you.